Okay, this is photosynthesis. My name is Radhika Gamaria 2.6.
My name is Gabby Fragoso, 2.7.
My name is Max Hill, 2.11.
Shem.
I'm Shem McCarthy, 2.21.
I'm Mark Alonzo, 2.2.
We're P.2G.1, and this is photosynthesis. So two water molecules are split. We're going to pretend this is the resulting electron. This electron travels through the ETC, through photosystem 2 to a chloroplast. It is then struck by a photon to become more energized. It then passes through an embedded protein to power the active transport of hydrogen ions into the thylakoid. By the way, everything's happening in the thylakoid at this moment.
And previously, Shem said that the dark circle here was a chloroplast. That is actually the chlorophyll.
Thank you. The electron then travels to photosystem 1 to another chlorophyll, is struck by an additional photon, and then exits the ETC to be used in the reduction of an NADPH molecule into NADP plus molecule to NADPH. Then the hydrogen ions built up in the thylakoid follows their concentration gradient through ATP synthase to power the production of 3 ATP molecules.
As shown here.
Moving on to light-independent reactions, which occur in the stroma. RUBP, with the aid of Rubisco, an enzyme, is fixed with carbon to become PGA. PGA then gains the energy/electrons from 6 NADPH and 6 FADH2
and 6 ATP.
6 ATP, sorry.
And
6
also previously said CO2 is fixed with the aid of Rubisco.
6 ATP is hydrolyzed into ADP, and the energy enters PGA. This transforms PGA into PGAL.
6 PGAL.
1 PGAL leaves the stomata to be used in glucose production, although 2 PGAL are needed, and the other 5 enter. The other 5 are then turned back into RUDP.
Okay, and then we come over to to glycolysis. It is upside down. Give us a second.
It's fine.
Okay, so glycolysis, we're going to pretend this area is glycolysis. Okay, coming off of light-independent reactions, or the dark cycle, so 2p-gal from the dark cycle, or glycolysis, are used to form one glucose molecule. This glucose molecule then comes into glycolysis, which takes place inside the cell, outside of the mitochondria, in the cytoplasm. It is then, the glucose molecule is then broken into 2-pyruvate using 2 ATP molecules, which are then hydrolyzed into 2 ADP molecules that then go to oxidative phosphorylation to be phosphorylized once again. From here, the pyruvate goes through the cycle, and eventually it makes 2 NADH molecules from NADP+, where these are the NADH molecules. And then 4 inorganic phosphate also enter into the cycle to phosphorylize 4 ATP molecules. And the net gain here from this cycle is 2 ATP, as 2 were used in the beginning and 4 were made at the end. The 2-pyruvate then move on to pyruvate oxidation, also known as the link reactions, which take place here in the mitochondria. The pyruvate from here are oxidized, and they lose a carboxyl group from this. the carboxyl group escapes through the cell and is diffused out in the form of CO2. And from here, the now reduced pyruvate molecule, which is now known as acetyl, binds with coenzyme A, acetyl-CoA. That moves on to the Krebs cycle. But when the pyruvate was oxidized and lost its electron, the electron went into an NAD plus molecule and made an NADH molecule, which goes to oxidative phosphorylation. And from here, we move on to the Krebs cycle.
Alright. Hello, my name is Mark and I'm doing the Krebs cycle. So first, acetyl-CoA enters the Krebs cycle, and the acetyl group bonds with oxaloacetate to form citrate. Citrate then breaks down into alpha-ketoglutamate, and in that breaking down, it releases CO2 and it oxidizes NAD plus to NAD, or reduces NAD plus to NADH, and one NADH is created there. Then we go on to alpha-ketoglutamate to succinate, which also releases CO2 and creates and reduces another NAD plus to NADH. Then from succinate to oxaloacetate, FADH, FAD plus becomes FADH2, and NAD plus becomes NADH, which means we get a net gain of 3 NADH, one FADH2, and one ATP is phosphorylized between alpha-ketoglutamate and citrate. So we have one ADP to ATP, three NADHs, and one FADH2, and then we go on to oxidative, or the ETs.
The orange is FADH. Yes? Yes?
Yeah.
Orange is FADH?
Yes.
Okay.
FADH2.
Okay, so the FADH2 and the NADH is oxidized to its forms of FAD and NAD+, and we're going to pretend that this is an electron. The electron then travels to the electron transport chain through embedded proteins, which allows a concentration gradient of hydrogen ions to move outside the embedded proteins, and this then is there's also oxygen inside the embedded protein channel,
Inside of the
mitochondrial
Matrix.
matrix, which then binds to oxygen and forms water. That's it.
And these are embedded proteins, by the way, and ignore the sun. This is the matrix, and this is our inner membrane space. So the hydrogen that's pumped into outside the inner membrane space is then uses ATP synthase and is flowing into the mitochondrial matrix via facilitated diffusion, and during so ADP and inorganic phosphate is then phosphorylized into ATP, and then ATP is produced during this.
How many?
And 2 ATP.
34 ATP.
Wait, 34 ATP.
Alright, my turn. So,
Finishing off, you're going to do fermentation.
fermentation, I'm back. Here in the cytoplasm after glycolysis in an anaerobic environment, which means there's no oxygen in the environment, we go into fermentation. In fermentation, we use it so that we can replenish our supply of NAD+, so that we can keep glycolysis, and so we can do glycolysis again, so we can still get that 2 ATP. And lactic acid is produced. just say.
No, we go
Lactic
through
acid.
lactic acid.
Yeah, we go through lactic acid, and other organisms go through alcohol or create alcohol instead of lactic acid. That's all.